inhibition of is a target for treating RA, have they looked at drugs like abatacept? So yes, so Dr. Wittenbeek, um, yeah. So what I know from my previous background is abatacept is an inhibitor of the co-stimulation between CD28 and CD8086, and they did try this drug in RA, but they could not pursue abatacept in this indication because of I guess there are severe side effects like lymphopenia and also they could not personalize the dose for different types of RA patients, and that is why abatacept was never approved for RA, but is being used off-label in this indication. However, abatacept is now used in like it is under studies, sorry, not being used, but it is under clinical trials for graft versus host disease and other transplant-related immunopathologies. So they're used in liver. I guess they are also in trials in liver transplant and kidney transplant.
Okay, that's interesting. Did you also see my other questions? Go to slide 31.
Okay. Yes.
Okay, so I say that on 2 of these 3 studies you're presenting, you clearly show methylation effects through stats on FOXP3, but you see that it's with IL-2 you're getting stat activation, blocking methylation, increasing FOXP3 expression, and then IL-6, IL-4, they go through stat 3 and 6 to activate it. They promote methylation of DNA methyltransferase and decrease FOXP3. So I was going to say, like, how would you control for something like this with these opposing actions of the interleukins specifically and these stats? How could you control for that? Because methylation is pretty general. It's a hard process to study. How are you going to control for those opposing effects? Or how could you control for those?
Right.
I'm having trouble hearing you if you're talking.
Yeah, I'm thinking, Dr.
Oh, okay,
yeah.
I'm sorry.
Yeah, I'm just trying to look at my slide and also think about your question. I'm also thinking about your question.
That's perfectly okay. You can take your time.
Yeah, it's all right. We can't see you.
Sorry.
Well, we used to see you. Let me see if I can see you. There you are. Okay.
So if I understood your question correctly, is like, do you mean to say that during an active viral infection, there will be some IL-4, IL-6 as well, and then you should also have IL-2, right, in the environment? And how do you control IL-6, IL-4, like prevent IL-6 and IL-4 signaling so that we get more regulated T cell expansion through IL-2? Is that your question, Dr. Wittenbeek?
Yeah, and I mean, because if they're opposing, you might get a neutralizing effect and see nothing, and you'll say, oh, nothing's working, but really it's the fact that they're just negating each other. So it's always something you have to think about, because methylation reactions are just challenging.
Right.
They're pretty general when you start assessing them, so you have to really control for the IL-2 and or IL-6 and 4. Right. And then even the stats. So I'm just asking in general, how would you control for this based on your work?
Right. So, like, my answer, like, I'm not prepared to answer this question, but then I'll try. What I've seen in some of the clinical trial studies is that they're using low dose IL-2, and when they use low dose, and they're using it as a preventive mechanism instead of a therapeutic, like as a prophylactic drug instead of therapeutic, and they prime the humans with low dose IL-2 so that the STAT5s they get activated. But at the same time, it also increases the expression of CD25 on regulatory T cells. Like when you just add sufficient low dose IL-2, and these IL-2 are not the endogenous IL-2; they are engineered to produce IL-2 muteins. Like one I know is coming from Amgen, and these IL-2 muteins they are biased for T regulatory cell expansion. And these T regulatory cells, and such T regulatory cells also release immunosuppressive cytokines that have effect on the pro-inflammatory cytokine signaling, like the IL-6 and IL-4.
Are you finished with your answer?
No, so I just wanted to answer it by saying that you have to somehow create an immunosuppressive environment transiently. For example, it depends on the indication that you are treating, the condition that you are treating. For example, if it is transplant, so post-transplant you want T cell response to go down, and you want an environment which is conducive for acceptance of the transplant. In that case, the clinical trial studies are testing low dose IL-2 just before transplant, or they're also trying to infuse regulatory T cells in these the recipients, like transplant recipients, so that they have a transient period of immunosuppression before and in that period, like the graft gets accepted by the recipient.
So Vivek, though, I think Paul is asking in the context of your experiments here, though.
Okay.
Like, if you end up in a scenario where you have some IL-2 and some IL-6 and some IL-4, and maybe those levels are different between neonates and adults, but assuming that perhaps you're going to have some kind of mix, how might that affect your results, and what might you do to sort of address that? Because she's getting at the idea that those are going to work against each other and might give you like just a flat line in your data when maybe there's a lot of activity there.
Right.
So like in the context of
Thank
your
you, perfect.
experiments, yeah, so focus on your experiments here.
So Dr. Wittenby, for that, in that case, like I would like you to refer, I'll refer you to our labs data that showed in neonates when we performed array analysis for different cytokines. We saw that IL-6 and IL-4 were not upregulated in neonates, and I don't know, like if we will actually have to control these factors in our model, especially in the neonates, because they were not expressing sufficient IL-6 or IL-4, and overall immune response that we saw in neonates was more of a suppressive kind and not an inflammatory environment.
You know, Vivek, I just have a comment about everything that's been said so far, and that is that in the neonatal brain, the secretions that T cells bring to the battlefield may be much more toxic than in adults. And so you need the Tregs to calm down those responses, and the side effect of that is that they may not be able to mount the appropriate immune response to the virus.
Right.
But on the other hand, if you have an animal surviving, but it's, you can't have neural stem cell division that's properly filling the brain with functional neurons, that animal will not be able to meet the challenges in its environment with the appropriate adaptive behavior. And so I think what evolution maybe has done is kind of push the system towards, kind of lowering the inflammatory milieu in neonates at the cost of losing some of the neonates to the infection, because the substances that the T cells bring into the brain are so toxic, In neonates, in adults, they don't seem to be that way, which is a very interesting thing to kind of explain if there is such a mechanism for why those secretions simply wouldn't have the same effects in adults. Because the adults in your slide 9, I think that's like kind of a key slide that you have up there that can answer a lot of questions. You have high T cell load, high T cell numbers in the adults, and you have low viral load, but the T cells are not toxic in the adults because they don't have the seizures, they don't have the brain dysfunction, they don't have the dysfunctional behavior. I don't know what you think about that, but it's kind of one comment.
Right. So in our array analysis, for different cytokines, we did see an increase in TNF alpha, particularly in neonates, not in adults. Possibly, and we also believe that some of these inflammatory cytokines are actually causing more toxicity than the T cell infiltrates themselves. And yes, then it makes complete sense. Like if IL-6 is also present, it's also present in the environment, in that milieu, it is possible that the damage that we are seeing in neonates is also because of some of these pro-inflammatory cytokines, and not directly because of the T cells.
Yeah, wherever the cytokines are coming from, maybe they're not from the T cells specifically, but that's sort of like an epiphenomenon. There are other cells that are also secreting other substances that are high in the adults but don't have toxic effects, because in the adults you have very few dividing cells, and the dividing cells that are going to become neurons in the brain, whereas the neonatal brain is still filled with, kind of neurogenesis still has to proceed appropriately in neonates. And so that's maybe one explanation for why there's so much sensitivity to the cytokines and other, reactive oxygen species and other things besides cytokines that immune cells will release.
Right. So I believe our first aim should, where we are trying to look at the cytokine profile in the brains of both the age groups, will inform us more about the IL-6 and the basically it will help us distinguish between the type of immune response that is activated in both adults and neonates. Adults, we know that it is more of a Th1 response, and in neonates, we don't know yet. We have evidence in the literature that suggests more Th2 kind of response, and across different conferences, what we are hearing is they have more Th2 kind of response. But somehow we do not believe in such such dynamics, like adults can purely have Th1 and neonates can purely have Th2 response. But definitely beyond T cells, there are some other factors as well that are responsible for neuropathology, and that is what we are trying to understand in our aim 1, where we're looking for the cytokine profile in the 2 age groups.
Can I ask a quick question?
Sure.
Adding to that, so in all cases, does the RNA lead to changes in the actual proteins and the cytokines? You're looking just at RNA profiles, but do all those RNAs create the cytokines? Because you got to be careful. You have to usually prove it with an ELISA.
Right. Yes, we should be. Actually, a good point. like once we like confirm this thing in our like using the RNA, we will have to do an ELISA test as well to confirm if these cytokines are actually getting expressed in these two age groups. Yes. So
I just had one extra question, just in general, just kind of I think epigenetics is fascinating. And I just ask because you're bringing it in and you're clearly going to have maternal influences. Do you think that is the epigenetics that you're looking at coming in from the maternal influences, or do you think they're just going to be happening through in your models and your treatments, or are you relying on the maternal influence, because that also should play a huge role?
yes.
And what do you know about maternal influences on epigenetics?
Right, right. So it is a good point, Dr. Wittenby, because what we have read in the literature that inside the womb, since we have a semi-allergenic environment, and we also have this mix of mother's immune system plus the immune system that is that the womb that the fetus has for itself, and how the fetus is protected from mother's immune system throughout the birth process is also important to understand, I guess, because there's one study where they showed that fetuses have their own immune environment, and it was actually the NK cells that acted as regulatory cells over there. But then they also saw increased regulatory T cells in the fetus compared in the fetal circulation compared to like mother's circulation. So possibly that is preventing the fetus from activated T cells in mother's immune system. And I don't know if that is getting inherited for some period of time after birth. And yes, so like fetal T cells are more, actually they are more prone to Treg differentiation than neonatal T cells again. And possibly neonates are getting some of these T cells from the parent. Also, I would like to highlight over here that in neonates, we still have thymus. The thymic involution has not yet happened. And thymus does generate Treg cells. And in adults, after the thymic involution, we do not see any natural regulatory T cells. And possibly there is a difference in the type of regulatory T cells that both neonates and adults have. And that is the whole idea, like we're planning to confirm at the TSDR level whether Treg cells in neonates are totally different compared to the adults. Like are they, and of course, like it would be interesting to consider consider this point whether it is getting inherited from mothers. Yeah.
In fact, I think Dr. Kavanaugh has a question in the chat. You want to scroll down and get to it, or would you like me to read it?
Has the studied age-dependent drop in Tregs past 4 months of age? Past 4 months of age. So if so, what was noted in older ages? So Dr. Kavanaugh, I like at the top of my mind, like I do not have any idea, like if there were studies that just compared like animals beyond 4 months through like adults.
Okay, I just wondered, what would happen at even older ages.
Right. Right. But then, like in my slide, if you see where I presented, I'll just share my screen again. Like there are several studies that try to compare neonates and adults, but comparison within like adolescents and adults, I've never seen that. Like possibly there are papers, but then I did not come across such papers.
Vivek, I have a few questions. Or are you, do you want to kind of expand on this a little bit, do this paper?
Yes.
Rahana, sorry, real quick.
Yeah, go ahead.
Can you hear me?
Yeah?
Yes.
Okay, I just want to follow up on Jane's question. So Jane, this is controversial, but in elderly animals, it looks like Tregs start accumulating again in peripheral tissues. And part of the concern is that in the elderly, you actually go back to having too many Tregs. And that's part of the reason that the elderly sort of struggle with other infections. But I don't really, I don't know how that staggers like across ages of mice. I wouldn't consider a 4-month-old mouse elderly per se. So I don't really know what would be going on at 4 months. But you might start expecting like a reversal of the trend that we saw from, you know, newborn to adult. And then from adult to elderly, you might start to see that flip.
Right, that makes sense. Okay, thank you.
Thank you, Dr.
It's a very interesting idea that like the most vulnerable periods of life are in the neonatal and, you know, like very old age, and that I would not have expected that T levels would be high in old age animals as a means of immunosuppression. That's quite surprising.
So most of the studies that were done to compare age-specific differences in regulatory T cell expansion, they considered either percentage of FOXP3 cells as the total percentage of either CD4 or lymphocytes, or they just considered the cell counts across different age groups. So what we plan to do here is, like, of course, we'll be calculating the frequencies as well. We'll be calculating the frequencies as well, because we are more interested in knowing the change in Treg when normalized against the weight of the tissue, because nobody has done that. Nobody is considering the neonatal brain and the size of the brain of neonate and size of the brain we have in adults. So what we plan to do is, like, we plan to count the number of Tregs and like this will be normalized against the weight of the brain.
You know, I would do it both ways. You guys always do it per cell, per 100,000 cells or something like that, right? But the weight of the tissue will be very subject to edema changes, and that's going to throw off some of the results of this formula. And the other thing that might change is the weight of the matrix. So I think the more important thing is as a fraction of cell number. What do you think? Maybe try it both ways.
Right. So one limitation of this method is, like, we are assuming that both the brains are like homogeneous when it comes to immune cell infiltrations.
Right.
Yeah.
This is always the problem, and you have to express it as a ratio, but you just kind of don't know. I mean, you can also show the denominator by itself to see if that's changing.
Right.
In, you know, as a way to kind of show that, you know, there is an effect or there isn't an effect on the ratio through changes in the denominator and not just the numerator.
Okay.
Are the students okay? Do they want to ask any questions? I have lots of questions, but I can always ask them later. So just want to see if some students want to ask some questions before the faculty have to go off.
like in the initial stages of like the neonates, the increased number of Tregs causes not inactivation of the CD4 and CD8, which causes which decreases the survival rate. But in the later part, when like if you are depleting Tregs and you are expecting that it would increase the survival. But my question is like how it can be like if you are depleting Tregs, then TCD4 cells and CD8 cells will be more, which will also cause inflammation because they also release cytokines. Like the cytotoxic T cells and helper T cells also release pro-inflammatory cytokines. So how would that inflammatory cytokines will be balanced? Like in your slide 38, you have shown that you expect that with the Tregs depletion, you will see decreased astrogliosis. But the activation of CD4 and CD8 will also cause inflammation, pro-inflammatory cytokine release. So you might not get that astrogliosis in the Tregs depletion as well. Similarly, with the axonal damage, excitotoxicity, you might not get this expected results because the activation of CD4 and CD8 also causes
Right. So that is one of our alternative outcome as well. Definitely, that suppression in Tregs will make all the rebound increase in T cells. But another important thing over here is we're not chronically depleting regulatory T cells in our model. This is just one single dose of CD25 monoclonal antibody, and this will be given before we start the study, so that we ensure Tregs are depleted when we started the study. And then we're not kind of chronically doing this in our body. I don't know if that answers your question.
Okay, then the next question, I have one more question. You were talking about the FOXP3 expression. I just want to know that is it really like it's only present in the Treg cells or it's also present in anywhere else in the body? And you mentioned a study in which this FOXP3 expression was deleted or knocked out, and then you saw that there was autoimmune disorder. So was that study like in vitro, or it was specific knockdown, or it was a global knockdown? Like, what's the role of FOXP3 other than Tregs?
So FOXP3 becomes very specific because FOC and FOXP is an entire family of transcription factors, and what we've seen, like, and also reported in the literature, is that FOXP3 expression, they always talked FOXP3 expression in terms of regulatory T cells. Like, you won't find any literature that says FOXP3 was expressed in some other cell as well.
Oh, be careful with a statement like that.
Yeah, like
Be real careful with a statement like that.
Correct, right. With caution, I would say that. But yeah, like I haven't at least like read a paper that suggests like FOXP3 expression in some other tissue, like possibly functions might vary, but for T cells, yes, FOXP3 expression in T cells is important to maintain the Treg phenotype. That is important. But like I have not come across any paper that says FOXP3 expression in some other cells. Maybe some other cells apart from immune cells. They might be doing some other functions over there because FOXP family is a ubiquitous family, definitely. But FOXP3's role in other cell types, we don't know yet.
And what about that study you were mentioning about that decrease in FOXP3 expression? Was it in vitro, like was it like global knockout, or was it specific knockout in which you saw the decrease of the FOXP3 causes autoimmune disorders?
So it was in vivo injections of like CD25 monoclonal antibodies. So when you give CD25 blockers, basically monoclonal antibodies that act on CD25 receptors, what happens is they've seen suppression in the Treg expansion. Not that like every Treg is depleted, but then the efficiency that they quote is more than 90% efficient. They were this technique is more than 90% efficient in depleting Treg cells. So most of the papers that I read where they tried this technique, they reach somewhere around 90 to 98% of Treg depletion.
So I wonder, it seems to be when you explain that FOXP3 gene, so you mentioned that it's kind of like a transcriptional factor. So what's the target for the FOXP3? You know what I mean?
Right. So
Can you repeat the question for the people who are
Sure.
on Zoom?
So Dr. Yong is asking
FOXP3
if
target
FOXP3 is responsible for a regulatory T cell phenotype, then and also if FOXP3 is a transcription factor, what role it has within the cell, like what all functions it is associated with. So the answer is FOXP3 every functions that a Treg does. Like I on the slide where I explained how Tregs inhibit CD4 and CD8 activation. So it has a role to play in every targets over there in during that interaction. So I'll just quickly bring that slide up. Yeah, on this. So FOXP3 expression will lead to increase in FOXP3 protein. FOXP3 is a FOXP3 transcription factor. FOXP3 expression will lead to increase in FOXP3 transcription. That has binding sites across the different genes, and it kind of increases the expression of those genes. For example, increase in immunosuppressive. Some genes might be responsible for increase in immunosuppressive cytokines like IL-10, IL-35. I do not know the exact mechanism with which they do that, but yeah, they increase. This leads to an increase in the cytokines. Then also some genes that are responsible for decrease in the interferon gamma themselves. So when interferon gamma is suppressed by Treg cells, the MHC expression on APCs, they go down. And then expression of some of the surface receptors also depend on the FOXP3 expression, FOXP3 transcription factor. So in the presence of FOXP3 transcription, cell surface expression of some of the factors like CTLA4. CTLA4 binds to CD8086, leading to inhibition. So and also in that case, expression of CD25 receptors. So there are multiple genes that get expressed when a FOXP3 transcription factor binds to their promoter regions, and that helps regulatory T cells to perform its immunosuppressive function.
Yeah, and I have another question on slide 12. So I think that's most important part of your message. So I wonder how the neonate can develop their immune system, like how fast or like how slow. So are there any possibility like their high mortality might be due to their immature immune system?
Immature immune system.
Yeah.
Yes. So that depends. What is the definition of when you say immature?
So for example, like
immune system?
For the immune system, they have to try to make like antibody or like a T cell from thymus or like other organs. So
it is totally possible, right? It's totally possible because say for example, if neonates, like when you say they do not have a mature immune system,
so they
possibly they do not have some of these factors. For example, sufficient expression of APCs might be responsible for the immaturity that you are talking about. Yeah. So yes, definitely, an immature immune response, or I would say immature immune system in neonates will definitely be the reason for suppressed T cell response. But then, what is causing that immaturity? Possibly regulatory T cells. We don't know. Possibly regulatory T cells also responsible for that immaturity. So,
Vivek, I have some questions about your optimization. Why did you not do that in neonates if they have so many Tregs? Why did you do that in adult brains? It would seem to make your life a bit harder, right? What was the reason?
right. So I would say that is because like a straight answer is like I lack those skills of you know. So I was trained. I'm still trained on adults and pediatric brains. I need some training on neonatal brains, which is our plan. Like,
Oh, okay.
yeah, that's the only reason like I did not start with them, and I wanted to start something really quick, and I had like adults in there, and another important reason is we would have in adults we usually see higher number of cells compared to neonates, and if there is any like say subtle difference, we'll be able to catch that in adults and possibly not in uninfected infants, neonates.
Okay, that kind of leads into my next question of the CD4 expression drop that you saw, and you were wondering what that was about.
Right.
What controls CD4 expression in general? Like, what are the factors? And sort of related to that, I was wondering what the function of CD25 was, because I don't remember you mentioning much about that.
Right. So CD4 expression, what I've read in the literature, it says CD4 is found after helper T cells like they start getting activated and form CD4 naive T cells. Actually, all these T cells are they develop in the thymus. The progenitor cells, they come from bone marrow. They go to thymus, and then all of these T cell types they mature in the thymus, and over there in thymus they pick up different roles, and then we have these different types of cells depending on the cytokines that are activated. For example, if you have interferon gamma in the surrounding, like the cells surrounding helper T cells, if you have interferon gamma, and you also have interleukin 12 in the proximity, the downstream signaling in these cells, like these naive cells, leads to a greater expression of CD4. And for CD8, you need paracrine functions from CD4. So CD4 releasing IL-2 also helps activate some of the CD8 naive cells. So these are the cells. These are the expressions that the T cells acquire during their development in the thymus.
And you think that the interferon gamma levels, which normally stimulate CD4, are lower in the brain than in the spleen?
It has to be, Dr. Leek. Like I don't know the answer to this, but then like logically speaking, it has to be that way because brain is considered to be immune privileged, and that is one reason even neurons do not have the MHC expressions. Like of course they in some circumstances they do start expressing the MHCs, but in general neurons do not have these MHC expressions that can interact with CD4 T cells. So I'm just assuming that we'll see less CD4 T cells in the brain, and we're also seeing that in our lab, like in the uninfected adults and in the neonates, we are seeing CD4 cells, but not not comparable to what we see when we infect these animals in the brain.
Okay, do any of the students have any questions? Any more questions? If not, I have a few sort of big picture questions. You had mentioned that the measles virus's only natural host is the human being, but doesn't it infect great apes too, or no?
You said apes?
Yep.
Okay.
Yes, as well.
But then, where I'm coming from is actually you can also infect mice without these mice having the receptors. But that is a chronic process where you keep injecting these mice with or exposing these mice to measles virus, and over a period of time, even mice. And there are different models that that are based on perpetual exposure of measles virus to mice. And I'm assuming the measles virus infection that you're referring to in apes is also because of those apes being perpetually exposed to measles virus environment, and not due to a recognized receptors that were like a receptor that was identified in them. So the three receptors that I was talking about, they were all identified in humans.
Okay,
So-
okay. So you're saying that apes are not a natural reservoir for measles virus, and that they don't have the right receptors. Is that what you're saying?
Yes.
Yeah, so Vivek, so
Sure.
you can infect primates experimentally. They are not natural hosts for the virus, but you can force them to be infected. They do have the receptors. They're really hard to infect.
Right.
But there are certain strains that work in primates, and that you can do in a lab setting. But you don't see measles in circulation in primates at all. It's not like a pathogen that's passed amongst primates. So you can do it experimentally.
Why do you think it's not found so much in the wild, the measles virus? What is it about humans? Is it only the receptor three-dimensional structure that sets us apart from other primates, and that's why we're sort of natural hosts, or is it something else?
It can also be it is possible, like I'm just thinking out loud because I don't know the answer for this, but
I think that's
it's
okay.
possible that the immune resilience in these wild animals is like higher, like it's more in them, and that is causing a complete clearance of virus, and not in humans. We're not able to clear the viruses completely, and that is what we are also seeing in our model as well. We're not able to clear the viruses completely. I don't know if that's the answer.
I was just thinking maybe population density would be the answer to that question, and it just hasn't kind of taken off in primate populations because the populations are not as dense
Right.
as the humans, that's all.
Right.
Any other questions from the students?
Yeah. Rachel has a question.
I have a question on this slide actually. I know that you mentioned harvesting the spleen, the brain, and the lymph nodes too. And even though the lymph nodes aren't on here, I was wondering if you had like an expectation for where they might fall in like regards to these values.
Right. So when I say lymph nodes, I'm referring to like peripheral system and not central. And these draining lymph nodes usually like they are the sites where the antigen presentation takes place, and from there the T cells migrate to the infected site. So like I don't have any experience working on with the lymph nodes, but the idea is to confirm if peripheral change in the regulatory T cell expansion or suppression of regulatory T cell expansion is causing neuropathological, like it's correlated with the neuropathological events happening in the brain, or peripherally induced. I did not introduce that topic this time, but then last year, like I showed, there are like two types of Tregs, and the peripherally induced ones. We are also interested in knowing if peripherally induced regulatory T cells are actually able to cross the blood-brain barrier, or they're actually tissue resident, brain resident T cells identified as regulatory T cells in the brain, or these peripherally induced T cells are crossing the blood-brain barrier. So we would like to see if that happens. Then I would consider the Treg cell like to be higher in lymph nodes, but it is also possible that like higher for neonates in the lymph nodes, but it is also possible that we may see less Tregs in an event where the Tregs have already migrated to the basically the activated Tregs have migrated to the tissue of the site of infection, and they're no longer the deposits. Because one such study had this thing, they compared spleen Tregs and lymph nodes, Tregs in the lymph nodes. What they found was migratory, most Tregs that had migratory receptors, like the chemotactic receptors on their surface, their population in the lymph nodes decreased because they've already migrated to the spleen and the other tissues. But what they saw was Tregs that were not capable of migrating their population was higher in the lymph nodes. So it depends on what are we looking at. So if you're looking at say CD69 positive or CXCL3 positive Treg cells that have these migratory functions, then yes, I'll find like less of them in the lymph nodes, more in the periphery, in the tissue, infected tissue, and less in the lymph node. But in the spleens, yes, I'll find definitely more Treg cells. Did I answer that? Yeah.
So you mentioned that there are other viruses also that can infect the CNS. I'm interested to know if the same or similar immunosuppressive Tregs are also responsible for the mortality in neonates.
So depends on what kind of virus you are talking about, because within the viruses as well, there are like multiple types of viruses depending on what kind of nucleic acid they have. If it is say single-stranded RNA or double-stranded DNA virus. So it depends on we're not like I've not read anything in other forms of viruses, but ours is a negative sense single-stranded RNA virus. And I've also seen this thing happening with like a recent paper that I was reading on HSV, herpes virus. I've seen this thing happening there. Like there was an increase in the Treg response in the neonates, and what they showed was T cell migration to brain, and the effector T cell migration to the brain, and the effector T cell migration, like even the effector T cell migration to spleen, kind of decline. So, and they had low CD8 T cells, lower granzyme production. So CD8 T cells are also responsible for granzyme production. Yeah, so that was encouraging actually, because we expect similar response in our model as well.
So I was just thinking if you could like translate your findings to other diseases.
But what most of these studies were done peripherally, none of them in the brain. So it's again question to ask, like, what was the reason they could not directly see them in the brain? Is it because there were not sufficient cells in the brain? They were not able to measure that. So that's what like we're thinking, because whatever they saw was in terms of either migration of T cells peripherally to the center. If they have these migratory receptors, then they consider, okay, they possibly migrating to the brain as well. So yeah, so our study will be purely will be measuring regulatory T cells in the brain and also in the periphery. So that, like, we are kind of trying to de-risk our studies. If it does not work in the brain, we want to see, like, if peripherally by peripherally inducing depleting Tregs, does it, like, changing anything in the brain?
A lot of the Tregs might be in the meninges because that's sort of like a way for them to be close to the brain without getting into the brain. That you should look at that because there's lots of immune cells that are hiding in the meninges around the brain. They're including what are known as border-associated macrophages. So they're all along the borders of the brain, but they don't go into the brain itself because it's too risky. At any rate, at this point, I was going to, let me check one thing here. I was going to call for another break so that we can start on the stats part after the break. Is that okay with everyone?
Sure.
Okay. All right, so let's take another 3 to 4 minute break and then we'll come back and we'll do the stats. Thank you very much, Vivek, for your complicated project. You know, and it's always very nice to kind of hear what your lab is doing. So I'll let everybody go, you know, chill for a bit and I'll set up for the stats part. Thank you. Yeah. Vivek, would you mind making me a co-host?
Sure. Like you are the co-host now.
Vivek, let me know when people are back. Okay, I've tried to share something on my screen. I don't know if the class can see it.
So we're just waiting for Asif to return.
Sure. Thank you for sending me these data, by the way.
Yes.
Gives us something. It's nice that it's the same thing that was discussed in class today, the same topic. Okay. You ready, Vivek? I heard some noise.
We're just talking. So we're still waiting for Asif.
It's hard for me to hear you. I think somebody will have to stand in the front.
So, Dr. Lee, we are still waiting for Asif.
Sure. Do you want to turn the camera up to the class so that I can see
Sure.
who's talking and
Yes.
it'll help me a little bit?
Sure. Can you see us?
I can only see the ceiling.
Oh, sorry.
Yeah, so I'm starting to see more people now. I can see, yeah, just a little bit further back would be great.
Okay.
Thank you. Perfect.
So
Right.
I think you won't be able to see Melanie, except Melanie, like you'll be able to see everyone, I guess.
Yeah, I can see more. I can see 4 people now.
Okay. And Asif is back.
Yeah, yeah, all right, great, okay. So, Vivek sent me these data today, and I think we will kind of continue what we were doing last week. I gave you a very brief background on the statistics, kind of the rationale behind what tests to use, and also I gave you a handout that describes a little bit what you can do as a workflow, okay? Now, let's follow that workflow and see if it works, because it doesn't always work, and then at that point, I want us to kind of come to a mutual decision as to what to do when this workflow doesn't work. So in order to go through the workflow, I'm going to start with Vivek's very nice PCR data, showing essentially, I think intuitively, a biologist would look at these data and say there's a definite sex difference there. The females have very high variation compared to the males in interferon gamma expression, and the expression in females is higher than the males, okay? I think all of us would kind of agree with that. But let's go back and see what tests to do in a very simple example like this, okay? So when you have your data loaded into GraphPad, the best way to start by loading the data into GraphPad is to put everything in columns. And then later on, you can put it in groups as necessary for the two-way ANOVAs, but really kind of the best way to start is to put everything in columns. Here we just have 2 groups, the measles virus infected females and males. And what you can start by doing is just start with the t-test, okay? And the reason for that is that it will tell you right away whether it's Gaussian and homoscedastic or not. So these are unpaired data because they're different sexes, and we're gonna start with this assumption, even though I know it's wrong, okay? This assumption that both populations have the same standard deviation is wrong. Females have more variation, but in order to test the Gaussian distribution, we need to have that clicked, okay? The other thing to point out here is that it's safer to go with the two-tailed recommended p-value testing. So that just tests the reason it's two-tailed is because if you look at the distribution of p-values as, a frequency distribution, you're accepting if there's going to be an increase or a decrease in the experimental group compared to the control group. If you choose a one-tailed p-value, then you would only accept as significant if it was increased or decreased, but you're not open to both, okay? And so usually the two-tailed p-value is used. The other thing to mention is that if you choose one-tailed, the p-value, the probability of getting a positive result or a significant result really becomes higher, because the p-value, when it's two-tailed, essentially it divides p is equal to 0.05 by 2. So now your p has to be lower than 0.025 in effect. So usually just kind of go for what's recommended in GraphPad, unless you have really good reason to suppose that it can only be one-tailed. It can only go in one direction. So this is all I have clicked so far. I don't know if you guys could see that part of it. Were you able to? Or was it only showing this screen? I don't know what you guys could see. Were you able to see my, when I had that dialog box out, what I was pointing to, was that visible to you guys? Yes? No?
Yes.
Okay, great. Well, now this should also be visible because this is surrounded by the green lines that you can see in Zoom. At any rate, so you have here an unpaired t-test. The f-test is what it uses to compare the variation across groups. And you can see that it failed the f-test. In other words, there's highly significant differences in the error bars. In the females have much higher variation than the males. The other thing that happened is that the Shapiro-Wilk test, which I'm going to highlight in yellow, showed that it also failed the normality test. So then you would be like, okay, if you look at the decision tree that I have printed out last week, if you guys still have that today, it's not Gaussian, it's not homoscedastic, so you cannot use the t-test. It's not Gaussian and heteroscedastic, so you can't use Welch's correction either. It's non-Gaussian and it's heteroscedastic. So really, it would be best not to use the Mann-Whitney U, but there are no other options, because and I've already done this to save some time, when I did a transformation of the interferon gamma data into log base 10, and then did a testing of the normality again, it's well, we can do that quickly. If you do this by t-test and just do the same thing all over again, okay? The problem that arises is that it doesn't pass the normality test, even though it passes the f-test. So the f-test to compare variations is no longer, there's no significant difference in variation across groups anymore. And the data are, I don't think they're graphed, but at any rate. So there's no significant difference in variance, but there is no bell-shaped distribution. So we have no choice in this case except to use the Mann-Whitney U, which Vivek has done here, and there is a significant difference between females and males. And I think anybody kind of looking at this would be like, yeah, I don't really care what the stats say. That's really a biological difference between males and females. And if the statistics can't resolve that, that's a fault of the statistics, because here it's very clear to me that there is variation. Even if you don't look at this number here, almost all of the other numbers, except for a few at the bottom, don't overlap with the males in the distribution. And so that would kind of indicate, to me at least, that the common sense would dictate that these are really different. Yeah, I have to stop share at this point. Do you guys have any questions for me about that? That's kind of, what you're left with is only really the Mann-Whitney U, because the other reason to use the Mann-Whitney U and not the t-test here is because in there, 1 second to explain that. So the distribution, when you look at the Man-Whitney U, it doesn't care whether it's bell-shaped or not, so that doesn't matter anymore. It does care a little bit if there is heterogeneity of variance, but in this case, there's heterogeneity of variance also because one group is all squished down to 0. There's almost no interferon gamma expression in the males that are infected with the measles virus, which I think is a really important observation. So really there's kind of no way of getting around this problem that you have that there will be variation, the standard deviation, right? And so you kind of stick to your guns and you say, well, you know, do you want to do a t-test even though the distributions are not bell-shaped? Probably not, because this is it's very bottom heavy, this distribution, and then there's a tail that goes up, so it's really not bell-shaped at all. If you look at the mean here, you don't have an equivalent number of points below the mean and above the mean, and so I would say that that assumption really is not met by these data, and I think Vivek did the right thing by going with the Man-Whitney U, although the Man-Whitney U doesn't really like heteroscedasticity or heterogeneity of variation, it's still the safer of the two. So that's kind of where I would decide to go with the Man-Whitney U as Vivek did. Do you guys have any questions about that? Okay, now I'm going to share Vivek's. Vivek, do you want to share it on your computer or so that everyone can see? Let's see, what am I sharing? I'm just sharing my whole screen here. I think, I don't know exactly what the best sharing situation is, but if you guys can all look up here, I think maybe that would be the most efficient thing for now. I think in the future though, when we do more of these statistics examples, and I'm in the classroom, I'd like to see all the students kind of share it through this, like log on to the Zoom. Like next week, when Yang, you come on, Yang, you have some of your graph pads ready, but don't worry about making it perfect for the class, okay?
Yep.
Like I felt bad because I think Vivek and Lauren spent some time finding like graphs for us and preparing graph pad files, and I was like, don't worry about that. So in the future, nobody really has to do that, okay? Just use what you have. It's okay if you have a ton of tests that you've run to kind of teach yourself what the tests say, you know, that's okay, it doesn't have to be a perfect file. So what Vivek has here is CD4 T cells and CD8 T cells and B cells. And the data, always kind of look at the data structure by graph before you decide kind of what to do, because intuitively you will understand a lot of what's going on by looking at the data distributions. So let's start with this one. Here, I'm gonna say, okay, that's pretty good in terms of a bell-shaped distribution for this group, for this group, for this group, and for this group. There's probably gonna be heterogeneity of variation though. And then you might ask yourself, is it okay to use an ANOVA if there's heteroscedasticity in this case? I would say if you want to look at a statistical interaction between the two variables, in this case, age is one variable, and the other variable is measles virus infected or control animals. In that case, you have to use a two-way ANOVA. So the results of the two-way ANOVA are not in here. I think we cleaned it out. Let's repeat that. Can you see this part of my, see this part of my,
Yes.
of my screen? Great, okay. All right, so you want to fit a full model because you need to look at the interaction between age and infection, okay? So fit the full model. You don't have repeated measures because it's not over time and the animals are not paired in any way. The column factor in this case is infection status. and the rho factor is age at infection. Is that, will you agree with that, Vivek?
So, got
And then we're going to do this one. Now, we talked a little bit about this choice before. How many families should it be? We were going to choose the 2 families as more conservative because, well, for multiple reasons. A, because it's more conservative. This is recommended 4 families, 1 per row, but we had some issues when we did that last time, including that the lines wouldn't all appear at the same time. Let's start with this. The options, now it can allow, okay, one problem with this option is that now you cannot correct for multiple comparisons. You're gonna have a hard time getting that past reviewers, okay? If you use Fisher's LSD test, that is the same thing as not correcting at all for multiple comparisons. So try to avoid the LSD test if you can. So here you have to choose the more conservative option, and then now you can correct for multiple comparisons. You can use the SIDAC if you'd like, or you can use the Bonferroni, whichever one. There's actually not a huge difference between the 2. And in this last dialog box, you want to choose the bottom 2 options. Don't worry about the QQ plots. That gives you tons of graphs. Don't worry about that for now. You want Spearman's rank correlation test for heterogeneity of variance or heteroscedasticity, and want to see if the data are bell-shaped. So then you just hit OK. And what it throws out at you is the following. Wait, can you guys see all this clearly? Okay, so it's back to this page. So there is an interaction between the 2. It's a very strong interaction. What that means at a theoretical level is that the response of the CD4 T cells to infection is highly dependent on age, okay? In other words, the 2 variables interact. And then each individual variable is also highly, highly significant. These are really wonderful data that you've sent us, Vivek. I mean, it's like really clean, beautiful data. The other thing to look at here is that you go down and it does not pass the test for heteroscedasticity, which is what we all predicted when we looked at the data, because some of the data points were really squished down, right? There's no way of getting around that. This is just truly the biological response in the uninfected animals is going to be really low. So I'm not sure we need to worry too much about that. I'm surprised that it didn't pass the normality test in a way, but actually when you go back and look at this, if you combine all 4 groups together, they're not normally distributed because 3 groups out of 4 are pretty much back at the bottom. So if you were to make a frequency graph of all data, all 4 groups all together, there would be too many animals that are close to 0 and just a few animals that are going up towards 20. And so it's not going to pass the normality test either. So then what can you do? Let's go and consult the workflow that we have. You have 2+ groups. They're non-Gaussian and heteroscedastic, so we could log transform if there are no zeros. Are there any zeros, Vivek? No. Nothing that is exactly 0. So you could log transform, for which you just go up here to transform normalize, and you hit OK. It's y equals log y that you want, and you hit OK, and then you get some negative values which look really kind of odd, but it's okay because you can go in and you can change that. In general, like I said last week, I really like the box and whisker plots, and I like to show all the data points in the box and whisker plots. And that's what it looks like. So just let's talk a little bit about this again. Here you have an effect. If I were to draw a line, maybe I can't draw a line, oops, not here. If I were to draw a line from here to here and make it red so that it's more visible, right? And then take that line and draw it like that, from here to here, on the median, I should say. you can see there might not be as strong an interaction in the log transform data because the lines are more close to being parallel. But what you see in these data is that infection increases CD4 T cells, and being an adult also really increases the CD4 T cell response to infection. So and the adults in general are a bit higher than the neonates. So there's three things happening, which is reflected here in this data table here. You have an interaction, the age of infection, and infection status are all highly significant. But let's see what happened after we transformed the data. For that, you have to go into the results section where the transformed data lie, and you re-hit analyze, and you do the two-way ANOVA again. Everything should pretty much be the same. I'm just going to leave that as we did before. I'm going to do that. Yeah, sorry, I do this always, so that you can actually correct for multiple comparisons. We'll use the Bonferroni for consistency and keep these two checked. Okay, so now what happens is that the interaction becomes weaker as we would have expected, okay? But the row factor, the rows are age and infection. The column factor, which is infection status, are both still highly significant. So there's not as strong an interaction in the log transform data. It still doesn't pass the normality test, but it's no longer heteroscedastic. So let's look at that. Again, it doesn't pass the normality test still. So that's annoying. Then you might ask, what will you do? Well, you go back to your statistics workflow. So even the log transformed does not meet the assumptions of parametric testing, which is a Gaussian distribution and homoscedasticity. So what are the options you could use? Well, one thing you could do is use the Kruskal-Wallis, and nobody would question you on that. But again, like all non-parametric testing, the Kruskal-Wallis and the MAVID-EU, they don't like heteroscedasticity, okay? So, what I would do in this case is I would say, okay, you could try the Kruskal-Wallis. The reviewers would really like that. You would have to transform or change these data into column format to do that, because the Kruskal-Wallis is really a one-way ANOVA. So with the Kruskal-Wallis, you can no longer test an interaction between the two variables. But when you go back and you look at your data, you're like, what's really striking about this, and I'm going to zoom in so that you can all see it, is that with the raw non-log transformed data, there's a huge change in terms of the adults are the ones that really mount a much more powerful CD4 T cell response compared to the neonates. And so the log transform data kind of hide that a little bit in not a good way. So I would say if I had to do this, and if I was going to report these data, you could say I'm going to do the parametric two-way ANOVA anyway, so that I have the power of looking at a statistical interaction between the two variables. When you go back to the two-way ANOVA, what do the p-values look like? The Shapiro-Wilk doesn't pass the normality test, so that's quite a low p-value. And this p-value for heteroscedasticity is also very low. I think one thing you could do in this example is you either use the Kruskal-Wallis or keep it with the parametric two-way ANOVA and just be kind of transparent in your method section. It's always good to have a really good statistical description section in the method section of your paper where you say, you're very clear about what you did. Like, we did parametric testing regardless of Gaussian distribution and heteroscedasticity. I remember years ago, I was on a PhD dissertation committee where Frank D'Amico, who's a statistician who worked a lot with Paula in the past, and he said it didn't matter. He would prefer always to do the two-way ANOVA and just be done with it, because what you can say to the reviewer is that the two-way ANOVA is very robust to the assumptions of the two-way ANOVA not being met. Okay, that's always something to remember about the ANOVAs, that they are not that vulnerable to these assumptions not being met. So, so I have to say that some of this is a little bit subjective, right? And that's where kind of people can get into a little bit trouble. That's why we have a workflow, but sometimes even after transformation, it doesn't adhere to the rules of parametric or even non-parametric testing. So then you're left with like the decision that you have to make as to what you're going to do. Remember when Muslim asked last week, what do you do if something else, if you don't follow this workflow and you get significant p-values? Now, I would say you should definitely start with the workflow. And, if you get p-values that are non-significant with the workflow working and p-values that are significant with that but that are not in the workflow, then you have a harder time kind of justifying it. When you can say to the reviewer, when we did the transformation, we still did not meet the assumption of parametric testing, but oh, by the way, where's that here? We did pass the heteroscedasticity test, and the important observation is that the interaction, the row factor and the column factor are all still significant. So one thing that we've done in our papers is kind of show data that are analyzed in multiple ways. So with and without log transformation, before and after, you can do that. Or you can say, the graph on the left was analyzed with a two-way ANOVA, but because it did not meet the assumptions of parametric testing in terms of bell-shaped distributions, we also did the Kruskal-Wallis and show them side by side. And then nobody can fault you for that because you've shown everything. I bet, your data are so nice, Vivek, that if you did the Kruskal-Wallis on this, it would still show you, as I look at this transform data or these data, the Kruskal-Wallis, which essentially ranks everything, will definitively show you that this is higher than this still. Maybe this won't be higher than this. Do you want to try that out now?
Sure.
Should I try it out, Vivek? What do you think?
Sure, Dr. Yes, Dr.
Okay, all right. Well, let's then squeeze this down together, because GraphPad does this strange thing of making you whoosh all the way to the side if you swipe a little bit too aggressively. So I'm going to do the following. I'm going to take this and start a column entry where I hit paste transpose, right? And I know from here that this was uninfected and this was infected. So this can just go here. This is neo-infected, am I right?
Yes.
And this is adult infected, right, Vivek? Is that correct?
Yes.
Okay. CD4 T cells, adult versus neonate column format. Okay, and here we can do the Kruskal-Wallis by going to one-way ANOVA and non-parametric testing, and then what should we choose? Well, it's not paired, so we leave that alone. We do the non-parametric test, and it only gives you the Kruskal-Wallis as a choice. So here, this is an important part here. Do you want to compare everything to everything here? What do you think? I'll let the class answer and then I'll tell you what I think. My lab can't answer this question, I'm not allowed. Because you guys already might know the answer to this. But the rest of you, what do you think? Do you want to compare? Do you want to try that?
Dr. I would do everything to everything. No.
Every other column.
I can't really hear you, Vivek. Can you repeat that?
So I would like to do, I don't know, what does it do when I say everything to everything? Like if I say everything to everything, will it kind of compare infected and the uninfected neonates as well?
it'll compare everything to everything, but if you do that, you have two variables. You have age at infection and you have infection status. You won't know which variable is leading to the difference if it's comparing, for example, PBS animals to in PBS neonates to adult infected animals. You won't know if it's because they're adult or they're measles virus infected that you see the difference. So, the more of the comparisons you choose, and these are called post hoc comparisons because you decide them after you look at the data, they're not a priori comparisons. But the more comparisons you do any place, like the one-way ANOVA, for example, if you have like 100 groups, you'll never get anything to be significant, and that's going to be a false negative, okay, which is a type 2 statistical error. Rather, what you want to do, here you can pre-select. You want to compare neonate to adult, that's fine. You want to compare neonate to neonate infected, that's fine because this is only differing by one variable. And then you want to compare adult to adult infected, that's all fine too. Is there anything that I've missed?
No.
I don't think so. Neonate to adult. But you don't want to compare adult to neonate infected because these are PBS animals, these are measles virus animals, and they differ also in age. And so you wouldn't know what to attribute it to. I don't think that would make sense. We want to keep it more like the two-way ANOVA structure, which automatically only compares across one variable if you do it correctly. So we'll just leave it at that, and I'm going to hit OK. And then the options, the dones should be selected because you always want to correct for multiple comparisons when you do statistical hypothesis testing. And then it doesn't let you do anything here because this is the Kruskal-Wallis, and the structure of the data in terms of a bell-shaped distribution doesn't matter as much. So the Kruskal-Wallis says overall there's a very significant main effect. That doesn't tell you which groups are being compared. It is not yet what's known as a pairwise comparison, okay? That's hidden in here. Here are the multiple comparisons. This is just overall, there's some significant change across the 4 groups. And it's a very big change. The multiple comparisons show you this. So you can see right away that the Kruskal-Wallis has very little power, okay? It gives you something that's an adjusted p-value of 0.0115, unlike the power of the two-way ANOVA. So if you go to the, if I insert a graph here, give me a second to insert a graph. I'll just do that, and then I'll just do that. That's how they like it in their lab, and so then I'm going to just go automatically add the pairwise comparisons, and it did it for the Kruskal-Wallis. And that's the only thing that's significant. Nothing else is significant. And, if you had to kind of, I just do this not because you can kind of just go and decide where you have the lowest p-values, but to make you understand that if you don't use parametric testing, your power to resolve a statistical difference really kind of goes down. As I look at these data, I'm not too worried about lack of bell-shaped distributions because this is pretty bell-shaped, this is pretty bell-shaped, this is okay, and that's not quite bell-shaped, but the lack of Gaussian distribution, it only happens when you combine across these different groups, but they are different biological groups, so why would you be surprised that there's different distributions when you combine them? They're so different that it's always going to be non-bell shaped that when you combine them. So that's another reason that I'd be okay with just sticking to the parametric two-way ANOVA for data like this. And you could show it with and without. You could show it as with the Kruskal-Wallis and with the two-way ANOVA side by side so that the reader can make the decision about kind of, what the sizes of the effects are with parametric versus non-parametric testing and take it from there. It's interesting because when you do the two-way ANOVA, is that graph, and you do the comparisons, it doesn't allow you because GraphPad is silly that way. That's very annoying that GraphPad can't do that, and I think it's because we have this clicked. If you do the, I'm not sure it can, I think we would have to, just for sake of this class, compare each, if you do this, that doesn't really give you much information, but we're just gonna do it because that's the only way that GraphPad can see, doesn't correct for multiple comparisons. It's really not good if GraphPad to do that. But if you do that just in an extra sheet, then what you can do is use that to throw down all of the lines to make it easier for you to draw the graph. But then you don't use these p-values because everything is being compared to everything, and that doesn't make sense. You go back to a different sheet and you do the two-way ANOVA properly. You just use this, graph where all the lines are already added, but you use the two-way ANOVA correctly. Okay, you make a duplicate of this, duplicate the current sheet, you do the two-way ANOVA again. And in this case, now you don't do something like that, but you do this and that. Options should be to correct. You always want to correct. And then you can do that. And then you use these p-values. Unfortunately, you have to manually add them. So if you really want to use a better graphing package, that's fine. There's other graphing software as well, but GraphPad is very easy to use. And what I like about it is that it gives you things that are recommended. Usually they make a lot of sense, not 100% of the time, but usually they make a lot of sense. Finally, let's look at these values here when you compared everything to everything as if there's only one variable, which is not the case here. You can see that this group was not significantly different from that group, but this group is significantly different from that group. Now, I still think that the neonatal viral infected animals have a true biological increase in CD4 T cells. It's just not as high as the adults, okay? So that's my interpretation of the data, regardless of what the stats here tell us. It's just that the adults have such a big response because everything is so small relative to that response, they're not going to become that significant, okay? That's the other problem with statistical testing is if you have 4 bars and one group is really through the roof and all the other bars have differences, because of that one group, nothing else will be significant next to itself. And that's another reason to mitigate that, you can log transform. Okay, because those are some of the kind of things that people struggle with a lot. Now I know that, with GraphPad, it's pretty easy to do all kinds of analyses, and they say, oh, don't do that, that's p-hacking. You really have to teach yourself what the tests show you by doing multiple types of analyses. Otherwise, you'll never learn how the different tests work. So don't feel bad about a GraphPad file that has a ton of types of tests on the left-hand side. It's normal to do that and to train yourself into am I comfortable losing a lot of power by going to the Kruskal-Wallis? I think if you're not comfortable with that and you want to leverage the power of the two-way ANOVA, but you still think that some people really may not like the fact that it's so heteroscedastic and so non-Gaussian, you can show it both ways and let the reader decide. I think that's a good option to do it. And that's all I have for you as far as this lecture. It's really strange to be here and lecture this way. I apologize for that. Sorry, my ankle is popping in and out of its place right now. So do you have any questions for me about Vivek's file? Yeah, Muslim.
Ma'am, I just want to ask that what is adjust p-value, ma'am? Like, is it
What is, I'm sorry, maybe can you speak a bit louder or come to the front?
adjust p-value? What is that, ma'am?
A what p-value? Adjusted p-value.
Adjusted p-value. What
Yeah, that's a good question. I don't know the answer to that question. For some tests, they show an adjusted p-value. For other tests, they show an exact p-value. It's really getting down into the weeds of statistics that I wouldn't be able to answer that. Whatever GraphPad gives, I mean, should you report it as adjusted versus exact? Maybe. You know, it's not a bad thing to have to report, but it gets very busy. A lot of papers in the really high impact factor journals, what they have is they have a supplementary table that goes on for many pages